My name's Wendy Mock. I work at Nelson Laboratories. I'm the packaging section leader. I have the privilege of participating on all three of these committees, the USP, the ASTMs, and the ISO groups. So there's a lot of exciting stuff going on in the packaging industry right now. And I'm going to take a short amount of time and share what those changes are within, obviously, confidentiality agreements and things like that. So let's get going. That way. Oh, you know what? There. I got it. So I'm going to talk about the changes that are going on in the industry, including both pharmaceuticals and medical devices, because both of them are changing significantly, and they're going to affect the packaging industry. So I'm going to talk a little bit about the update to Chapter 1207 that's specific to pharmaceuticals, and then I'm going to jump into some ASTMs and some ISO standards. But first I want to talk about what's the purpose of changing the methods. Why do we want to change these methods? As our technology is evolving, our test methods are getting a lot better. We're getting more sensitive, we're getting better capabilities. It is improving faster than the test methods, so the test methods have to evolve along with them. But why we want to do this is more important. It's because as these methodologies are getting more and more sensitive, we're finding smaller and smaller defects, and deciding whether these defects are actually life-threatening is very important. And so as we go through this, we're going to talk a little bit about that, and some of these leaks that are being detected are so small, you have to determine whether or not this is applicable, and is it worth saying, well, we need to recall all of our product, because the after effect of that is that now we're going to start getting things like drug shortages, which will affect life. And we don't want to do that, because patient safety is our most important goal of all of us here today. So how are the standards made? It depends on where you sell your product and what your regulatory body is after. If you're looking to follow the USP requirements, they're a scientific nonprofit organization that sets standards specifically for drugs. And these are enforceable by the FDA if they're considered a chapter of 1,000 or lower. So they will come and audit you to these chapters, and if you comply, you'll be in great shape. The ASTM methods are volunteers. The USP is nominations, whereas the ASTM is volunteers. Everybody comes together with their industry experience, and we create these standards to help out based on our experience in the industry. And then the ISO is exactly similar, except now you're branching out into Europe and different countries. So each of these, though, has the same end goal, and that is to bring modern technology and experience and science to our packaging standards so that the end use is appropriate and safe for patients. So what are our successes in the packaging industry? So as I mentioned, the biggest change right now that just released on August 1st is the 1207 chapter in the USP. It took a long time to get this rewritten, is the right word, because it used to be only like 3 pages long. And now it is in excess of 40 pages and has 4 separate chapters dealing with different areas of testing. The important part to note about Chapter 1207, though, is it is specific to non-porous packaging intended for sterile pharmaceutical products. So if you have a product that is not non-porous, this chapter doesn't apply to you. So it's specific to things like vials and syringes, flexible non-porous packages. Those types of things will apply to this chapter. It includes details and recommendations on the selection of validation It includes things on leak testing methodology, all sorts of really cool stuff. It actually will go out there and talk about some of the methods, why they're really good for your tests. So let's get into a little bit of them. In the first chapter, 1207, the key concept that they really push in this chapter is that packaging integrity is a key component of the entire product life cycle. It's not just a section like we've kind of seen it in the ISO standards. You test for integrity, you test for strength, you test for microbial barrier. This is taking the approach of this is part of the entire life cycle. So that is also the introduction to this term called leaks of concern. What type of a defect is actually going to affect your product quality? Is the defect too small that your product quality isn't going to be affected and it's okay? So it goes into a very strong discussion about what's considered a leak of concern. It also introduces this term called maximum allowable leakage limit, because if you have to define what a leak of concern is, how are you going to get there? Well, you have to use these mols. And what it is defining what that leak size is that is acceptable for a given product line. And so they will go into talking about a specific table that ties to leak sizes, which is also tied to gas rates, and then all these different methods, and we'll get into that. It's also important to understand that integrity is assured if packaging allows no leakage greater than the mol. So smaller, we really don't have to be concerned about, but defining that is where the difficult part comes in. It also goes into developing through the 3 phases, and this is actually very common. It's in most of the documents. You go through your design verification, you look at your manufacturing, and then you look at your shelf stability assessment. It also will show you some guidance on how to select and develop and validate those leak test methods. So there's one thing that's important. And when you're looking at those leaks of concerns or mols, it's ideal to understand that, so we're dealing with non-porous packaging here, right? That's what this standard is designed for. So if I have a non-porous package, it's important not to get confused with permeation, because everything will permeate. There's no such thing as something that can't permeate. But leaking is an intentional defect caused by an event, whereas permeation is a gas flowing through a solid barrier. Permeation is defined as inconsequential as far as product quality is concerned. But an intentional defect is where the leaks of concern come in. So as you are looking at your test method selection, it goes through an awesome list of the types of categories that you need to look at. What are the contents and what are the design? Are you working with a rigid or flexible material? Are you working with porous or non-porous? Because if you're working with porous, then this doesn't even apply to you, right? You're going to a different section, or I'm sorry, a different standard. What's your closure type? If you're looking at a mated surface, that's going to be different than if you're using a welded surface. Those are going to have different qualities. Also, how many times do you access the product? Is it a single use or is it multi-dose? So again, that mol is coming in there. What's your maximum allowable leak tolerance? And then obviously you're going to use physiochemical or microbiological tests to prove the integrity of your methods. It goes into some really good versions out there. It starts introducing these terms called deterministic or probabilistic. And there's preferences for deterministic methods over probabilistic. That's going to be a change in the industry, especially with pharmaceuticals. because when we get into the slide, you're going to start seeing that some of these test methods that we've been doing every single day as part of routine are no longer preferred. And so what do you do if your test method's on the non-preferred list? So we'll talk about that. And then what's the method outcome? Do you have quantitative data, qualitative data? So this is where they get into the definitions for deterministic versus probabilistic methods. Deterministic are going to give you predictable results, but probabilistic are based on events to create that result. You're looking at a physical chemical endpoint versus a random outcome. Just all these different types of things. Another important one is there's almost no sample prep when you're using deterministic methods. There are a lot of, you just put it in the equipment, it analyzes it, and it takes and gives you a reading. It's very non-destructive. And if you have a leak, you can actually do a lot of root cause analysis. Whereas if you jump into the probabilistic methods, there's a lot of sample prep required, and unfortunately, they usually end up being destructive test methods, and root cause analysis when you have a failure is very difficult. So this is going to be a really big change for the pharmaceutical industry because of this classification. This section, 1202, goes into it in much, much more detail, talking about we prefer this over this. And then it will actually pair each test method to a leak size. So, for example, it will say that a 1 micron hole is commonly detected with this test method. So, things like that. So, somebody's actually gone out, and obviously it has a disclaimer, because the second that somebody says that this method will give you this sensitivity, because that's kind of what it is, right? If they're tying a leak size to a test method, it's like establishing a sensitivity. And so, but it does have its disclaimer that this can be different depending on what you're testing, what your suitability of your product is with your container, because all of that will have an influence on the test methods. It's very important to say, and I have these, they're direct quotes. They are that deterministic methods are capable of detecting leaks at clearly defined and predictable detection limits. So they're defined. Not so defined when you're looking at probabilistic. It's inherent. They're also the best chosen method when outcomes demand a specific approach. So these are very important quotes from. The probabilistics are very challenging to design. You'll frequently get issues with the test results, interference with suitability, problems with positive and negative controls, things like that will pop up in the probabilistic methods, whereas the deterministics, again, here's your value, pass or fail. So here's some examples of the preferred, and they do use that term, which is very strong, preferred deterministic tests. So if you have a liquid inside a vial, you would be looking at using a high voltage leak detection test. It just analyzes the electric through the liquid, comes back with a value. If it's not the value that's set with the control, you're going to have a problem. It's going to fail. Pressure decay has been around for a long time. We have mass extraction. A laser-based headspace is becoming a new technology. Well, it's not a new technology, but it's becoming very strong and prominent now because it's so customizable to different types of packaging configurations. You can use vials, you can use auto-injectors, you can use syringes, and it just aims that laser right at your product, analyzes the headspace. It can look for carbon, oxygen, or pressure, depending on what lasers you're using, and it will tell you really exciting stuff, like if you have a vial with a septum, What they're finding out now, this is one of those things where we're learning more as we go through science, is that as vials go through airplanes, those septums are actually shrinking and allowing gas exchanges in high altitudes. And then when you come back, they're back to their normal sizes. So you had a leak, but you'll never detect it when you're at your testing phase. So this methodology is now set up to design and will check the pressures inside. And so if you've had that leak as you go through the altitudes, it will detect the pressure change and show that you have an issue. So that's an example of these sensitivities and capabilities that are developing, and we're trying to catch up with them. So it's really exciting stuff. The probabilistic methods, you'll notice these have been around for a long time. You have the bubble emission test. Take your package, you put it under the fluid, you look for air escaping. You have the bacterial immersion. This one, you literally have to manipulate your product to contain not your drug, but actual growth media, so that if it leaks, the organism will migrate into the vial and grow the organism. So you have to manipulate your sample. That's where that sample prep issue comes in. Liquid tracer dye immersion tests. We're finding a lot of issues with suitabilities of the dye interacting with vials in the product, that they're not compatible, that even recently we just discovered that a lot of people are moving to putting these little capillary tubes in their devices and then exposing them to this liquid tracer gas or liquid tracer fluid. And what's happening is we've identified that over time, if you don't test, like say within a few hours, because when you puncture those capillary tubes in, the pressure pushes some of the product up into the tube. And so now your product is interacting with the environment and is actually solidifying in these microcapillaries, and you're getting false negatives. So there's going to be some research and data on even using these microcapillaries as suitability type detection limits. So that's kind of why these are not preferred methods, because they're very prone to having issues. So the next chapter, which is the last chapter, it's also the shortest chapter because pretty much it just references all the ASTMs. And the important part about this chapter is that the seal quality and leak tests work together to create package integrity. It's not just one, you have to have them both. And so it references lots of different standards, including the burst, the peel. The last one is a torque retention test. So these are all nothing new. It's just interesting that they've incorporated these medical device methods now into the pharmaceutical drug testing technology. So it's the beginning of a merger between the medical devices and the pharmaceuticals. So it'll be interesting to see where that goes in general. So now we're going to get into the ASTMs. That was the PDA, the USP. The ASTMs have had a ton of revisions the last year. This one is the ASTM F1608. It's what's called an FDA consensus standard. If you are doing flexible material that's porous, you would do this test. And it's FDA approved. And it changed recently to include a couple things. Historically, the only method of extracting the organisms, and the way that it works is you expose your material to organisms. And after it's exposed, we take your material and we put a filter underneath it, and we extract the organisms off this filter. And we look for how many materials or organisms got through your material. And so the only way to extract that organism in the past was to stick it in a blender. And I kid you not, it is exactly like a margarita blender. You stick your piece of material in there, press the button, and it chops it all up. And that's the extraction method. So now it's been revised to allow you to do different microbiological extraction methods. You can use glass beads, any of those, as long as you validate what method you use to extract the organism. So that's a really good step in the right direction, because blending is very variable. Try saying that 10 times. So you'll extract the same piece of material 4 times in 4 different blenders, and you'll get 4 different results. Whereas now we've moved to glass beads, and so that's like more of an impaction type of removal, and that is much more reproducible and repeatable. There's also been some changes to the diagram. Somewhere along the lines, half of it got chopped off, and we ended up going back and finding the very original diagram that was written by the FDA, and that's been incorporated back. But there's still this big issue to be dealt with on this test method, and if it doesn't get dealt with, it's going to have to go away. And that's the F1608 nebulizer. The nebulizer is no longer made. It's very specific. It's a DeVilbiss 40, and you can't buy it from a manufacturer. So you're going to have to deal with the fact that this needs to be either replaced or the methodology needs to go away. So that is to come the future. ASTM 2097 just recently published in June. And there was a lot of changes in this one. We now incorporated the biocompatibility into the flow diagram. If you know this one, it's got a really nice, this is how you meet this requirement, this is how you meet this requirement, and it sends you directly to the standards. So it's incorporated the biocompatibility. It also has added in a air permeance Gurley test. So if you have non-porous materials, you now have a test option rather than doing a whole package aerosol challenge. You have something for non-porous materials, and it's a very fast, very quick test. So there's 3 new oxygen transmission standards that have been incorporated. They added in the seal peel for round cups, and then the non-porous dye migration method has finally been added in, and now you have 2 options for climactic stressing to meet your distribution. Yes?
Previously validated products with this old version get affected or impacted by this change?
All of the old methods are still in there. Yep, these are just the new items. So they've, nope. It's just even more options. As those technologies are evolving, we're incorporating those. So it's really good. D4169 had a revision in June also. It seems like everything revised in June with the ASTMs. We've changed the assurance levels to power spectral density levels. So instead of assurance levels 1, 2, 3, we're now low, medium, high. And then they also doubled the frequency options. So that's a very technical discussion. If you're interested in it, I did bring the standard. We can take a look at it one-on-one that way. The same question. Okay, so this one has actually gotten easier. So because the profiles were more rigorous, they were based on leaf springs. You know what leaf springs are in vehicles? Now everything's air ride, right? So you're overexposing your product. So this is much better. And there's a lot more details to this one, and you could do an hour long on the changes to this one, and it's still going. So they're going to hit rail and air. So it's exciting stuff. So what else is coming, right? That's a lot of stuff that changed, and it's only September. We have 4 more months, and we have lots of stuff coming still. We're continuing to work on updating our peel test method. I don't know if you guys have done peel testing, but when you send trays in with lids, they're really hard to cut the coupons. And if you don't have people that know what they're doing, what'll happen is you'll get delamination in your results because of nicks on the sides of the coupons from the plastic not being cut properly. And so if we can come up with a method that will allow you not to cut out those coupons, but actually use the tray and separate the Tyvek while it's in its whole form, that'll make things a lot easier for everybody, right? So we're going to look at that. And then there's an ongoing evaluation about the difference between pounds per inch versus newtons per millimeter. So how do you, seems like you'd just be able to mathematically switch them over, but there's been some questions regarding that. Is it really 1 for 1? So there's going to be a round robin on that, or there was. So ASTM 2638, if you guys heard of this method, this is a new method that's out to replace F1608. It's called a surrogate F1608. And the way that it works is it uses a piece of your material, and we aerosolize particles on one side of your material, looking for the penetration of the particles through the material. And the output is what's called a Pmax value, or maximum penetration value. This is going to get revised this year because we've done a lot of testing with it now, and so the standard was written and not too many people did it, and we have a lot of test data now, and so we're going to do a few changes. Currently the standard states that you should start with a high titer and move to a low titer, which has actually been blinding the sample. So if you have all these pores and I bombard it with 10 to the 6 particles, I'm going to start plugging up some of those holes, right? And then as I get to a lower and lower titer, they're not going to get through because those holes aren't there anymore. So we're switching that to start from low concentrations and move towards high concentrations. Additionally, we are looking at incorporating what's considered a mesh screen. And right now, the way that it works is once you apply the titer, it actually ends up sucking the material down into the device. And so you actually will get blinding underneath because it's set against the holder rather than allowing the material and the particles to flow through. So using a mesh screen to support it will actually give a better value. If you're interested, I added Jane's information. I can give that to you at her request. She's in charge of this work item that is ongoing and is always looking for people to participate and read and contribute. So if you're interested in that, I can give you that information after. So the next item is the 2096 bubble emission test. I told you that's been out there forever, right? It's time to review. The round robin needs to be redone because it was only done on one material combination. So we need to look at incorporating probably non-porous materials, the effects of that. There's been several clarifications being made, like making sure that you don't overpressurize porous materials, things like that. This is a naughty standard. This is a German standard that they are currently in the middle of doing round robin testing. It is, the only reason that it is still around is because people in Europe and China want to pass-fail tests. They don't want you to give them a number and then say, okay, you rationalize what an acceptable number is. This will do that, unfortunately, but the science behind it is not there. So it will be interesting to see where this method goes in the future. Hopefully nowhere, but I doubt that. Another one that we're working on, Dan, I asked Dan if we could put this in. It's a very large document. We're looking at a guidance document regarding the validation of packaging testing. So if you're interested in participating in that, I can get you Dan's information. Is in the very beginning stages. It just went out for its initial review now, so it's a good time to get involved if you're interested. It's very, very math driven. So another thing that we'll have to start looking at is the surfactant listed in Triton X100 is being moved to the substance of high concern. So it's been identified as something that has disruptive properties to the endocrine system. So as people become aware of this, it's going to start getting banned. We've already received letters from some companies that are asking if this is used as part of your solution. It might not go anywhere because laboratory use versus, device use or something is completely different. There's not really much chance of us introducing it, but when you're looking at its impact in the environment, everybody impacts the environment, right? So there may be the future opportunity for identifying an alternative surfactant that won't be on that list. Additionally, ISO 11607 is under revision. We're looking at possibly incorporating conditioning requirements above. The thought is that if you use conditioning other than what the standard is, you need to be able to justify and rationalize it. It sounds like people out there are just skipping it, which I can say that they are just skipping it, and there's no rationalization as to why. Additionally, a reiteration again of why you're using your microbial barrier testing. What's your acceptance criteria? Don't just do it to do it. So, as you saw, there's many changes in all those different types of arenas for testing the packaging. The next few years are going to bring advances. It's exciting to see what new technology is coming and how that's going to affect packaging. But again, it's going to be important to remember that as we develop this technology, we have to be realists and find out what that acceptable leak limit is that will have harmful effects on our product rather than just saying it failed. So, any questions? Sweet. Cool. Thank you. Appreciate it.